That was an awesome. That was an awesome introduction. Thank you.
Of course. Welcome.
Cool. So I'm really excited to get a chance to talk with you. I love talking with students. I love talking about this topic generally. And I love it because usually students have a unique perspective, and you tend to come up with really awesome questions. And so I'd encourage you to go ahead and ask questions throughout the talk. I might not know the answer, but I'll try to take a stab at it. And anyway, we can have a discussion. So yeah, today I want to give just like a really basic overview on what the traditional drug development process looks like. I'm not sure this is ever like explained in any sort of formal setting necessarily. And so that's also why I kind of like to go through this. Like, what does it actually mean to work at a pharmaceutical company? What is drug development? Um, so we'll do just like a few slides overview on what that is. And then I wanted to talk about Alzheimer's disease and use it as a case study for what drug discovery has traditionally looked like. And then I wanted to go over this paper at a very high level. So this is a paper from 2023 from a group in the UK and Denmark that wanted to look for new drug targets for Alzheimer's disease. This is not a paper that I was associated with or anybody at Recursion, but it uses a very, very similar approach to what we were trying to do. And so kind of an interesting thing when you're working at a company, you can't really always talk about exactly what you're doing until you can license it. Or patent something. So there's a bit of a barrier to what I can talk about with you guys, which that's maybe a downside of working in industry. We can go into that as the talk goes on. Um, yeah, so then at the end, I just wanted to then talk about a little bit what we were trying to do at Recursion. And then finally, I wanted to have some time at the end about my own career path. It has not been traditional, but it's been great. Yeah. So let's jump into it. Also, is the sound quality okay? Y'all can hear me all right? Sweet. Okay, so I think there was another paper that I had sent out that we're not going to dive into deeply here, but I thought it was a nice like synopsis of like what the drug discovery process is. Nice little introduction. Um, and so basically you can split the process into these four main parts. This first part is what we're going to spend the most time talking about, the drug discovery process. And this is a part where you get a lot of failure actually. And actually it's better if you fail more here versus in the later stages, because the later you go, it's more expensive and you start getting into humans. So after the drug discovery process, you have your preclinical phase. This is where you start to do as many experiments in the lab as possible to rule out anything that could be toxic, rule out anything that could just be non-effective, right? So you try to increase your chances of something that's going to be as effective in people as possible and as safe as possible. And there's certain tricks to do that. After you get to the preclinical phase, you ask the FDA if you can start dosing in people. There's a whole process. They're very stringent, which is a good thing. You want them to be stringent. They give you the go-ahead, then you start going into clinical trials. You've probably heard of like phase 1, phase 2, phase 3 clinical trials. These different phases do slightly different things. Usually phase 1 is where you're just dosing in normal, healthy people actually, and you're doing different doses depending on a person's particular weight or their particular like physical characteristics. This is just to make sure, okay, in like a healthy population, minimal chance for any like bad damage. Is there a healthy, safe dose that you can use? Once you've figured out your healthy, safe dose, then you can start moving into like your patient population. So for Alzheimer's disease, that would be Alzheimer's patients. And then you have some readouts. This could be something like cognitive tests. This could be blood draws. If there's something in the blood that you know you can measure for that tells you if the drug is working properly. This could also be brain scans. It depends on what particular indication or disease you're going after. Finally, phase 3 is just sort of like narrowing everything down, figuring out what your dose is going to be for your final drug, what your recommended like dosing regimen is going to be, right? Like, do you have to give infusions once a month or once every 6 months? You have a bunch of, regulatory people checking all of this out, making sure everything is safe. You've got enough data to make a good conclusion. And finally, you have this phase of regulatory approval. Once it gets past here and it actually gets to market, doctors can start prescribing this. It doesn't end there. So there's still this post-market surveillance. So you just monitor how the population is taking your drug. Are there any adverse events that are starting to pop up that you hadn't accounted for during like clinical trials or doing regulatory approval? And, companies can, pull something off market if they have to, or, there's steps that people could take at this point. So this is a long process. This has historically taken over 10 years because a lot of compounds that have made it past a certain point will fail. Maybe they'll fail in phase 1. Maybe there's something that they find is too toxic. So maybe it's giving people some stomach troubles or something at a certain level. And so they have to cut it. They have to stop and say, no, let's go back to the drawing board. Let's find something else that might work better. And so kind of the idea is if you can weed out the bad compounds as early as possible, then you can make these later stages run more smoothly, more efficiently, hopefully more quickly and safely. And maybe you can actually accelerate these last 3 columns here. So now let's go back and focus in on what that early drug discovery process actually looks like then. And we can start thinking about, okay, what are some things that we can do here that can help accelerate those later stages? So there's this target identification and validation. So first, what do you want your compound or your drug to actually hit? So say that from this giant body of literature from different researchers that have studied this receptor, that you want to inhibit receptor X. And then you make your case as to why you think that's the receptor that you want to design a drug against. And so you do this sort of phase of like assay development. Okay, so how are you going to screen a lot of compounds or a lot of different chemicals to find the right inhibitor? And so maybe from the previous work that everybody had done on this receptor that that overactivation of the receptor leads to a lipid accumulation in some cell type, let's say astrocytes. Therefore, okay, we can do a screen for astrocytes and check to see if a compound decreases this lipid accumulation. Now, of course, you have to make sure, okay, that lipid accumulation is actually linked to disease. You have to make a convincing case that it's actually causing the problem that you see in patients. But that's one way that people like to model disease in cells for this kind of thing. And then you have this lead generation. So that means what's your lead compound? You've generated a lead compound. So you've found one, you've screened maybe 10,000 compounds, and you found one that inhibits the lipid accumulation in astrocytes really well. Say you only need like 1 picomolar concentration. It's very, very small. You only need a little bit of it, and it does a fantastic job bringing that lipid like level in the cell back to a more normal level. But maybe it has some properties that aren't that great. The chemists have told you, oh, based on the structure of this compound, we don't think it's actually going to get through the blood-brain barrier. So how is it going to help? So the chemists have to make some tweaks on that compound. And then you, as the biologist, have to do some experiments to then test the compounds that they give back to you. And you kind of iterate and iterate until you narrow that down to some group series of compounds that you want to move forward into the preclinical stage. So that's kind of the big overview. But that like target ID, that's kind of a big thing. How do you know what to actually target? That's a big, major question. And how do you know that the assay that you're running is actually representing disease? How can you make sure that that's true? That's where we have to rely on a lot of basic research, and a lot of basic research that's done with a focus on a disease or a patient population. So historically, people have used molecular biology, pathophysiology, and genetics. What do I mean by that? I mean that, so we'll see in the example of Alzheimer's disease, maybe you know that a certain population that gets this disease, they always have a mutation in a particular gene. Right? And so, okay, you can reasonably assume that it's either that gene is directly causing the disease, or maybe it's doing something that's affecting some other pathways downstream that are actually causing the disease. Especially in neuroscience, we rely heavily on pathophysiology. And what does that mean? That kind of means like, what does the disease look like? So like, for Alzheimer's disease, what does the brain of an AD patient tend to look like? And that can help you kind of narrow down, okay, what's some potential targets you can go after. And that's basically what I just said. Yeah, so are there any questions up to this point? We can wait for questions at the end, maybe, or I don't know if there's a question that comes up, maybe you can put it in the chat. I'm fine with that. Yeah, so before we hop into Alzheimer's disease, I just wanted to explain what are these types of screens that people do in this target identification phase of drug discovery. There are genetic screens, and then there's compound screens. So a genetic screen, usually it's one by one you knock out, or you can also overexpress each gene in the genome in a particular cell type. And you run those cells that have had one gene knocked out, you run them through an assay. And then that assay hopefully will help you identify genes that, when turned off or on, appear to rescue a disease phenotype. So first of all, you have to have a model of the disease, though. And this just tells you what the gene is. It doesn't leave you with any compounds you could actually turn into drugs. So then people do these compound screens. So you would run thousands and thousands of compounds through an assay. Ideally, you'd narrow it down to, gosh, like under 100 would be great, but maybe a couple hundred. And these will be the starting point for chemists to optimize. But for this, usually you need to know what your target is. And so there are some limitations here. So now we'll jump into see Alzheimer's disease and how this kind of process has been applied to Alzheimer's. So first of all, as you're all probably very well, Alzheimer's is a devastating neurodegenerative disease. It's the largest neurodegenerative disease. I think it's AD, and then Parkinson's disease, and then I think ALS are the big ones. As the population over the age of 65 increases, so will the incidence of neurodegenerative diseases. But until quite recently, there hadn't been any disease-modifying drugs out there. And actually, some might argue there still aren't. It's a little controversial. So Alzheimer's disease was first characterized by Alois Alzheimer over 100 years ago now. He described his patient, Augusta Dieter, who exhibited these signs of dementia. At her death, they took her brain at autopsy, and he had been developing these different staining techniques to look at brains of different patients. And what he noticed was this striking phenotype, the pathophysiology, which were these plaques that existed outside of the cells extracellularly. And then you notice these intracellular tangles. They look like these dark flames. They're flame shaped because they're actually filling up pyramidal neurons. And so his findings actually dominated a lot of the field for a very long time. Interestingly, he also had described these lipid accumulations in glial cells, so non-neuronal cells. This kind of fell off the radar of the entire field until about 10, 15 years ago. Maybe that's because not a lot was known about glia. There weren't really a lot of methods to look at lipids in a routine way until quite recently. But these are the three pathophysiological hallmarks of Alzheimer's disease that he described himself. Researchers later found that the plaques were composed of this peptide called amyloid beta, and that the neurofibrillary tangles were composed of this misfolded tau protein, a cytoskeletal protein. The lipid accumulations, we're still figuring that out. And so I think it was in the 80s and 90s, molecular biology and genetics techniques identified that there were mutations in amyloid precursor protein and the enzymes that produce amyloid beta from this precursor protein. These enzymes are called gamma secretase and beta secretase, or BASE. And so this is very interesting, very compelling evidence. Gosh, there's mutations here that lead to this overproduction of A beta that we know are in patients that get Alzheimer's disease. We also know that amyloid beta happens to be accumulated in these plaques, that this must be related. And so the field for a long time was very focused on, all right, let's make therapeutics that get rid of amyloid plaques. And then some folks were also focused on making therapeutics that get rid of tau tangles. So things that prevent the proteins from misfolding and accumulating in the first place, or make inhibitors to the enzymes that overproduce amyloid beta, for whatever reason, they're making too much. And we know, at least in vitro, when you add this amyloid beta, when you add the misfolded tau, it kills the neurons in the dish. So it seemed very, very likely that, hey, this is what you want to target. This is what we need to make drugs against. This is going to help people. And so, of course, that's not the case. Gamma secretase inhibitors, for example, were kind of notoriously terrible. Even beta secretase inhibitors, they weren't working. They were having adverse effects. Yeah, just failure after failure after failure. And then more recently, there's this aducanumab that maybe you've heard about in the news. This is actually an antibody, or they call it a biologic. It's an antibody that pharmaceutical companies will make, and then they actually just give this antibody to people in the form of an infusion. It does decrease amyloid plaques in the brain. It does decrease amyloid beta somewhat. It does not appear to be helping with the cognitive deficits that you see in patients, though, at least not that much. However, the FDA has gone ahead and said it's fine. Companies can give these antibodies. There's another company that makes a different antibody called lecanemab. Yeah, so, and then just by the way, also anti-tau therapies have also not been working. So there's a couple of reasons that you could think of that could explain why these aren't working. People for a long time thought, oh, maybe we're just not identifying the correct patient population. Maybe we're dosing patients with these drugs after the amyloid beta has already killed all of the neurons. And so there's some thought, like if you can catch patients earlier, maybe the therapies will be more effective. But maybe also targeting amyloid is not the way to go to treat Alzheimer's disease. I think in the last 10 years, there's been more of an acceptance in the field that, hey, maybe we can find alternative targets. I think people still acknowledge the role of amyloid beta in at least initiating the disease. There's a very good body of evidence that suggests that's still the case, but maybe clearing amyloid beta is not the right way to go for therapy. So it's sort of like back to the drawing board. If we're not going to be targeting amyloid beta or anything that makes amyloid beta, what are we going to be targeting? And so this is where we'll start to go back to those principles about drug discovery. First of all, what's your assay that can be used to screen? And so that's where this paper comes in. They are using this assay called cell painting. This is also what we use at Recursion, actually. So this is a way to visualize cells in a fluorescent microscope and visualize different parts of the cell. So there's a stain for the nucleus, there's a stain for the nucleolus, there's a stain for endosomes, for mitochondria, for the actin cytoskeleton. It can very easily, very quickly, and cheaply show you different parts of a cell that otherwise would be very hard to see. And so this paper in particular, they wanted to look at cells, wild type neurons, and they wanted to look at neurons that were knocked out for a gene that has been implicated in a previous genetic screen for being associated with Alzheimer's disease. This is sortilin 1. And people have known for a long time that neurons in AD patients also tend to have these enlarged endosomes. And it turns out when you knock out this gene called sortilin, you also get an enlarged endosomal phenotype that you can see fairly easily. And so they reasoned here, we can generate cells that are knocked out for sortilin. We can see this enlarged endosomal phenotype. We think this is really important and associated with the disease progression because it'll mess up the neuron's function. And so let's try to find some compounds that will make our sortilin 1 knockout neurons look more like healthy wild type neurons. So we'll find some compounds that will rescue this disease phenotype using this cell painting assay. So they did this and they generated a lot of data, a lot of imaging data, and then they used AI, machine learning methods to analyze all of those images. And they identified some compounds that ameliorated the disease phenotype. And this is kind of the same thing that we do at Recursion. So just to start off, they went through and knocked out the sortilin in their neurons, and they just show by Western blot, yes, we knocked it out. This is how they knocked it out in here. They just eliminated this particular sequence. And so they then checked the knockout neurons and compared them with the wild type neurons. And to do this comparison, they stained them for DAPI, so nuclear stain, but then also for these two markers that should be on these neural progenitor cells. And I should mention, I always thought it was very interesting that they used neuroprogenitor cells and not differentiated neurons. Probably the NPCs were easier to work with. And I wonder if they struggled with the differentiated neurons because they're just harder to work with. So that's sort of, I think, for me, one caveat of their study. But they looked at the NPCs expression of nestin and SOX2. This is what the wild type healthy neurons look like. And then the sortilin knockouts. Yeah, they're expressing it. They look a little wonky though, don't they? I don't know. They look a little different to me. And interestingly, they had a little bit less, they had less of the SOX2. That's right. They had less of the SOX2 marker. They didn't really elaborate on that. Maybe they're just reasoning that that's part of the disease phenotype. But yeah. So next they asked the question, okay, can we use cell paint on these? Because if we can't put these disease model cells through our assay, then we can't do our assay. And so the punchline is yes, they were able to cell paint the sortilin knockout. Again, there's some interesting, maybe slight differences here. There's maybe like a slight increase in this concanavalin A, the ER marker. maybe a slight increase in this nucleoli marker. Hard to tell if there are any differences in this actin cytoskeleton marker. Maybe some differences in this mitochondria. It's maybe less punctate in the sortilin knockouts, but anyway, they showed that yes, we can reliably cell paint these. So they were able to knock out, create the disease model. They were able to run the disease model through their assay. And so the next step was, can we use our AI and machine learning methods to actually detect phenotypic differences between these cells when we take images of them with the cell painting? Because if they can't use their machine learning model, then they also can't do their assay, right? And the punchline is yes, they were able to use PCA and hierarchical clustering to reveal quantifiable morphological differences between the wild type cells and the knockout cells. And so finally, they did their screen. So they screened with this library of a couple hundred compounds. So what is this library of compounds? This was an FDA-approved library. These are drugs that are already on the market and available, already have been dosed in people. And so a lot has already been known about these compounds. There are, different libraries that are out there. This is relatively small. A couple hundred is not that big at all. There are other libraries that exist that have thousands or tens of thousands or even hundreds of thousands of compounds, a lot of which there's not a lot known about them besides their structure, and a lot of, libraries that have like novel chemical matter, right? So these are all known. There's nothing new about them. So they screened through, and with their machine learning technology, they were able to identify actually of those, like the hundreds, they narrowed it down to 16 compounds that they found rescued this phenotypic change in the disease cells. Interestingly, most of them grouped in a similar pathway as well. That little bit is important, right? Because if they found that the group of compounds that seemed to be effective were all from like different pathways or like a smattering of different pathways, that could indicate some randomness and that maybe things aren't exactly working the way that they think they should. The fact that it's all of these compounds were coming from a similar pathway means that, hey, maybe they're actually targeting something that's biologically relevant that is actually helping the cells. Next, so sortilin A is just the protein that's made by the gene sortilin that they've knocked out. So they did this pathway analysis, or this analysis of protein interactors with the sortilin A in the NPCs in particular that they had. And they found that these protein interactors are involved in endocytosis. That's a great sign, actually, right? Because this phenotype associated with the sortilin knockout is enlarged endosomes. And so this all kind of makes sense. Again, it's like if the protein interactors that they found in their disease model or in the NPCs, if it was something different, it might indicate that there's something wrong with the system or like their experiments aren't quite working out. But next, they looked at what the known targets of these compounds were from this library, identified in the screen, and they just double checked, okay, the known targets of these compounds, are these targets actually expressed in the NPCs? Because if they weren't, then that would also be a big red flag, right? That things weren't working. But they confirmed by RNA-seq, yes, the known targets of these compounds are expressed in the NPCs. So probably the compound is working through these targets to help fix the phenotype. And then finally, they did another kind of pathway analysis, and they found that protein interactors with the identified compounds and the known pathways are also expressed in the NPCs. And they did this experimentally. This wasn't just using databases that were already available. So it really seemed like, hey, these are real targets that are getting hit by these compounds that they identified. These could actually be leads for a drug discovery campaign. Oh yeah, sorry, one more thing. They analyzed the compound structures against known compound target structures in MPCs, and they discovered that these compound structures were hitting potential targets that were known to be involved in AD, like base and gamma secretase, interestingly. And it has been hypothesized that the secretases might actually be overproducing amyloid beta due to differences in endocytosis or deficits in endocytosis. And that's why they're overproducing A beta. And so this all kind of made sense and fit into this kind of model of what they think might be happening in disease. Great. So are there any questions about the paper I can answer at this point? We can also just wait till the end.
Sorry,
yeah.
can you hear me? All right,
Yes.
I kind of have a question. Like, if it was shown that there was randomness within like the and they weren't grouped in similar pathways for like the compounds, where would they go from there?
Yeah, they'd probably start over. So they might, so what we do at Recursion, I think what you're supposed to do is first check your computational model and make sure that your model, your algorithm that you're using is actually correct. So we've done this kind of thing on different cell types. And a model that's generated to analyze images from, let's say, HUVEC cells, so like endothelial cells, that model is not going to work well in a neuronal model or a different cell type that has a different morphology, right? And that kind of makes sense. Like a computer algorithm that detects changes in little flat cells that are kind of like fried egg shaped, probably it's not going to be that good at detecting differences in like neurite length, right? Or like dendritic spines, or like something that's like a very, very different morphology. And so I bet if they had found some randomness in their data, they probably would have started with their computational models first. If they were still getting some randomness in their data, they'd probably go back to like doing QC on their disease model, making sure the staining was correct, the microscope image acquisition was fine, that kind of thing. Yeah.
Thank you.
Cool. Any other questions? All right. So this is all well and good, but this is all still starting from like a known gene, right? And so maybe there's some bias there. It's already based on, what people have found in the literature, in a kind of a biased way. So at Recursion, we asked the question, how can we find new disease targets that no one else has uncovered before? Or how can we identify targets in as non-biased a way as possible? And so now I have these fancy Recursion slides that were approved by our legal team to show you. There's this disclaimer that I have to show you. It's all quite silly. There's nothing proprietary in here, I promise you. So this is just an illustration, a traditional target-based screen where you start with a single target and you screen 1 million compounds, right? But you're still only going after one gene or one gene product. So we want to know, how can you do both at the same time? So how can you do that kind of genetic screen, but also at the same time do a big compound screen? And how can you do it in a cell type specific way? Because what you find in like an endothelial cell might not be applicable to a microglial cell or an astrocyte or a neuron. And so trying to do. We're trying to shrink this traditional drug discovery pipeline over at these later parts by expanding this first part as much as possible, right? So we want to fail quick. We want to fail early. We want to fail a lot of compounds sooner rather than later. And so we're hoping that that will getting better compounds earlier on through that preclinical development phase and through the clinical phase. And so that is it for me. Now I just wanted to go through my own career path. Like I said, it wasn't traditional at all. I knew in high school that I liked science, but I really had no idea where to start. I had a lot of really great teachers that believed in me a lot and helped encourage me to go to try different things. I had this awesome opportunity to work as a lab assistant at UCSF. I loved the lab environment. I was literally like cleaning glassware and like restocking pipette tips. But then eventually they got me like doing more fun, interesting things. They even had me like managing a mouse colony. I don't even understand why they did that. I loved it. It was a great experience. I got to do that and defer a year before I went to college. Went to UCSF or UC Davis rather. It was really difficult, really, really hard. And kind of questioned a lot, like, oh gosh, what do I really want to do? I still kind of thought I wanted to do something neuroscience and cell biology, but wasn't really sure what to do. So I took some more time off before deciding if I wanted to go to grad school or not. Worked as a lab tech at a place at UC Davis. And again, it was great. It was a wonderful experience. I loved being in the lab. I loved doing experiments. And so I thought, no, yeah, this is great. I got this. I can totally do this. I zeroed in on like, hey, cell biology of Alzheimer's disease. That seems interesting. I did a literature search for people that were working on different things. I was kind of interested in this amyloid hypothesis and beta secretase. And so I found the lab of Gopal Thanakaran at the University of Chicago. And as you're all probably very well aware, grad school is a very humbling experience. I was convinced I was a total idiot. Wasn't sure why I wasn't able to just publish papers all the time. It's hard and it can be a little arbitrary, and there can be factors that are outside of your control, no matter how hard you work at it. And so that kind of made me second guess things. Throughout my whole time in grad school, I loved having students in the lab, like I mentioned before. It was like a lot of fun, a lot of, really interested students. And so I thought, well, maybe I could do something like teaching, like a liberal arts kind of thing. I got this job at the Neurodegeneration Consortium, which is like, it was like an academic environment, working on these very drug discovery questions that are usually done in the pharmaceutical industry. And I thought 100%, no, teaching is what I want to do. I want to be a professor so bad. And then I landed the senior scientist position at Recursion. I also just really love the drug discovery process, though. It's like a fun puzzle to try to figure out, okay, what target do you want to hit? How are you going to design an experiment to figure out if your target is getting hit? And then there's this sort of satisfying, like, practical application of the work that you're doing. You're not just generating knowledge, you're actually driving towards getting a compound that could eventually be dosed into people and help people. I still think, both like basic research obviously is so important. You can't do drug discovery without the basic research. But the pharmaceutical industry is also kind of fun for that. So I listed this chart at the end. We don't have to go through this point by point, but I just wanted to show, some similarities and differences across academia and industry. Again, because I'm not sure it's ever like explicitly stated necessarily, maybe more and more in recent years. So what do I do all day? I still do the same thing that I do had done in labs in academia, run experiments, design experiments, analyze the data. It's all quite similar. It's just kind of your output is a little bit different. But yeah, it's a lot of fun. So I think that's it. So can I answer any questions? Anybody have questions about the paper that we went over, or drug discovery generally speaking, or I'm happy to answer career path questions as well.
Yeah, I have a general question about, it's kind of up the alley, like, what does a day in your life look like when you're working at one of these companies? And especially like in your role of senior scientist, do you do much of like the making of culture, stuff like that? Or is it mostly like you're planning and researching what could possibly be a target?
Yeah, perfect question. So it kind of depends also on the specific role that you're at and at a company. And I just learned this recently. There's actually two different career paths at companies. There's like the management track and then an individual contributor track. Individual contributor is going to be more closely aligned with what you would do in an academic lab. And so that's actually what I'm on. And so I'm a scientist. I design experiments and I run them in the lab. I culture cells. I am thawing microglia tomorrow to run some assays on them. It's quite similar. The assays that I'm running are immunofluorescent, right? Like I'm staining for different markers. I'm also going to be taking some media and measuring, secreted cytokines from the cells. My colleagues are going to be thawing neurons, and they're going to be doing this really cool technology called MEA, where it's in a 96 well plate format, and they can actually detect electrical impulses in a 96 well plate. I don't even understand how it works, but it seems really cool. And so, yeah, that's what we do on that scientist track. And so the way it goes, it's kind of silly. It's like research associate, and then scientist, and then senior scientist, and then principal scientist. And then if you're on the management track, you more become like a director. And so you probably spend a lot less time in lab. In fact, you might not even step foot in the lab. If you're in that management track or director kind of position, you're more just kind of like checking in with the scientists running the work and making sure that kind of the logistics in the background are working smoothly, so that the organization as a whole are, they're accomplishing their mission. Because my small little experiments that I do, that's just one small part of what is happening at Recursion. So the directors and the management folks, they kind of have the more bird's eye view of what's all going on and making sure everything's all coordinated properly. So, good question.
Do you still want to teach?
I do. It's hard to not. I love it. I don't know. I talk about it with my husband a lot. Like, but I think at this point, the thing with industry is that you're actually de-incentivized from publishing papers. And that's kind of a problem with trying to come from industry and jump back into academia. Because I think a lot of universities want to see that you've been productive and that you've been sharing your research across the field. And to do that, really the main way is to publish. But we're de-incentivized to do that in industry because we don't want to leak out trade secrets, right? And so it's very rare to have the go ahead from legal to be able to publish something. Usually if somebody's published and they work at a company, it's because the thing that they published on failed and it's not going to go to market. And so no matter how great it sounds, it probably they're publishing it because they can't make a drug out of it. So.
I was wondering what the collaboration process looks like in industry in general. Like, are there times when you are collaborating with other labs or academia, like your universities? Yeah. And how does that work in terms of like doing that while also keeping your industry secrets and everything?
Yeah, that's legal. You have to just ask legal before you communicate with anybody. It's yeah, so here at Recursion, we have a collaboration with a couple companies. There's Bayer, the pharmaceutical company from Germany, and then I personally am working on a collaboration with Roche and Genentech. And so the meetings that we have regularly with them, like I have to clear my slides and what I'm going to say, whatever my messages are, I have to clear it with a team here at Recursion first to make sure I'm not saying the wrong thing, I'm not making us look bad, and I'm not sharing any other secrets from other like departments at Recursion. So there's other people at Recursion that aren't working on those collaborations, and I'm basically not allowed to talk science with them at all. It's a little weird. It was different than a company. It was still part of the University of Texas and MD Anderson Cancer Center. So it was academia, and we were able to Houston, but we also worked at a lab at MIT and then another lab at Mount Sinai. We had regular lab meetings with them where, you know, just by Zoom, we shared our data slides with them all the time. Still, you know, universities still have a legal department. They still want to make sure that if there's anything patentable, that they protect certain information, right? I think a famous example of this is CRISPR between UC Berkeley and the Broad Institute, right? There was some fighting there about turn CRISPR into like a commercialized thing. Right, so that a little bit of that still exists in academia, but yeah.
So I don't know if you're allowed to disclose this, but I'm not too familiar with like, I don't know how the drug system works. So do you like when a pharmaceutical company comes out with a like a brand kind of drug, it usually costs more. Is there a reason why the generic comes out years later?
Yeah, there is. There's I forget what it's called. It's some sort of copyright thing. And basically like long ago, the government decided that something could be patented or copyrighted for some period of time. That's patented, I think. And it's like 10 years or something like that. And then after that, you know, the way that you make your drug can be freely available to the public. And then that's why, you know, generics can come out, and it's often, yeah, way more affordable, probably closer to the actual manufacturing cost versus the ridiculous markup that you see initially. Yeah, it's sort of unfortunate that it has to take so much time to get to that, but
So it just comes down to copywriting. That's insane.
You talked about how at Recursion you guys are trying to start like failing early more in the drug discovery process. How do you ensure that you aren't failing too early and like giving up on something before you really know all of its capabilities?
That's a really great question. And it's really, really pertinent. So what we're trying to do right now, what I'm directly working on, without giving too much details, is trying to come up with, frankly, like our disease model before we even get to our big screens. And it's been a struggle to come up with an accurate disease model, actually. Or it's not even like a disease model, right? Because we're going to be identifying hopefully genes that are associated with disease. Just coming up with like a model system. And you know, it's possible that could be a situation where we fail too early, right? If it's been very difficult, and you know, it's possible that it's going to be too difficult to where Recursion doesn't want to allocate the resources to it anymore. And so I think at that point, it just comes down to how much money do you have to spend on it for some period of time. Because, you know, you only have, it's finite, right? And so at some point you just have to say, you know, if by this period of time, which we think is reasonable to make some progress, if by this period of time we're still not able to do it, we're just going to cut it and move on. That doesn't feel very satisfying as a scientist because you still have that open question of like, oh, but what if I tried this other way? Maybe that could work, but that could be endless. And so in industry generally, not just at Recursion, there's this kind of mindset of like, look, like at some point you just have to cut the cord and you just have to say, you know, we can't spend time on this anymore. Let's go turn our attention to these other things that are maybe a little bit easier and more tractable.
We got a question in the chat from Vince. I'm not able to see it.
Are you allowed to publish once those chemicals enter the public domain? I think we're allowed to publish after we've patented. Oh, I see what you're saying. Yeah. After that patent's expired, probably is what you're saying. Yeah, I think we are. I think we're able to show the chemical structure, because that's another thing we're not allowed to show is the chemical structure, right? Because a very smart chemist could see a chemical structure and probably deduce how to make it. And so we're not allowed to share the chemical structure because that could show a competitor how to make a large amount of that. And so, yeah, we'd have to wait until after that time period before we could share it.
What made you choose private industry over academia?
Yeah, it's a great question. I think for me, honestly, it was actually Recursion specifically and actually not just industry. Like Recursion's promise of using AI to help accelerate this drug discovery process was really appealing. So before I had found Recursion, when I was still at the MDC, we had gone through this exercise of trying to identify new drug targets that we could work on. And so we were a small group of 10 people. And we each chose 4 different targets. We read through the literature and we each chose our favorite 4 genes. And then we went through this exercise of for each gene, making a slide and saying, okay, what is the gene? Do we want to inhibit it? Do we want to somehow like increase its activity actually? What do we think that would do? What would an assay be that we could run to check for compound efficacy? Are there compounds already out there? What are the companies that are working on? Right? It was an exhausting exercise. And we went through all of these targets, 40 of them in one day, and we were all so exhausted. And we tried to rank these. So we didn't want to do all 40. We wanted to choose like 5 of them, but we all came with our bias still, right? Like we all, had these biased reasons why we wanted to work on the targets that we chose. And then I read about, Recursion has this technology that they've developed here that eliminates that bias. And that was really, really appealing to me. And it seemed like it could be a really powerful way to get new drugs for Alzheimer's disease and other neurodegenerative diseases. So that made me choose Recursion specifically. But it's a real tight, close one with liberal arts colleges and teaching. I could easily still see myself jumping back into that.
That was something I thought interesting, just reading the article, like specifically the mention of machine learning. I was wondering if you could talk at all any more than you have about using AI.
Well, I can't, not just because of legal reasons, because I'm definitely not a computer scientist at all. I'm definitely the biologist. I'm in lab, like validating with what's coming out of the algorithms. So yeah, it's kind of a black box to me, and I think legal would want to keep it that way too.
I'm sorry. I don't know if this is gonna make sense, but do you have any conclusions as to why drug therapies that are reducing the AV aggregates are not targeting cognitive deficits?
Yeah, absolutely. Some theories that are out there are that, once you see the accumulation of A beta, you're already too late. the A beta has already done its damage. You've already seen some neuronal cell death, and you're just never going to come back from it. So then it becomes a question of how do you catch your patient population early enough? And it doesn't have to be with an amyloid beta clearing therapy. It could be with any therapy that's going after Alzheimer's disease, right? Because how are you going to catch these patients until they get the cognitive decline? And at that point, you know, how can you be sure that things aren't too late? And so that's where, you know, more careful understanding of the disease etiology is really important. We think that this burst of amyloid beta is an initiating event, right? There's actually some evidence to suggest that there's something that happens with astrocytes that kicks things off. And then microglia get all activated. And then you start to see some tau accumulation in neurons. And it's this tau accumulation that is actually correlating the best with the cognitive decline that you see in the patient. But then by then, maybe you're too late. And so, right, how do you identify your patients early enough? So what kind of screening assay can you have people do to be able to catch or predict like when you're going to see this progression of things happen? Is that going to be like test for amyloid beta in the blood? Or is there some other marker that's associated with astrocytes or microglia that you can test for instead? Is there some brain imaging assay that you can run? These are all things that people are looking into. So, but it's probably not going to be as simple as just clearing plaques. By then it's probably too late. And the plaques themselves probably actually aren't the ones responsible for the cognitive decline.
I was wondering if you have noticed any impact of AI, like the cell painting technique or like other algorithms on general job security, I guess, as a researcher, or
Oh, yeah.
like have that in mind at all?
Yeah, great question. We talk about that a lot at Recursion. Like, are we just like creating these things going to replace us? No, they're not going to replace us. They're not that smart. The computer scientists think they're so smart and they're making these amazing things. They're not that smart. You don't need biologists. You need a human brain. The human brain is too awesome. All right. Unfortunately, I'm going to have to get going, but this was great. I just wanted to offer if anybody had any other questions after this, please feel free to reach out. I think Oak has my email address. So I'd be happy to talk more.
Thank you so much, Dr. Fernandez. That was great.
Thank you.
Thanks a lot.